Learning objective include structure and function of flagella and also motility of a bacteria by flagella. Flagella, the word flagella basically is a plural of flagellum. This here is a singular. Here, when we talk more than one flagella, it's a long filament structure that propels bacteria. Right here, you could see this is a long filament thread-like structure that is used for motility or movement of bacteria. Flagellum consists of a protein called flagellin. It has three parts. There's a filament, there's a hook, and there's a basal body. In this picture, what you see is a flagellum of a gram-negative bacterium. Gram-negative bacterium has inner cell membrane, and then there is an outer cell membrane also. Between the two is a sandwich of a peptidoglycan layer. The flagellum in gram-negative bacterium, as I mentioned in the previous slide, it has a filament, which is a thread-like structure. There is a hook, and there's a basal body. The basal body in gram-negative bacterium consists of 4 rings. 2 are embedded in the inner membrane, and the rest 2, they are in the cell wall, in the periplasmic space, and also part of it is in the outer membrane. If you compare this with the gram-positive bacterium, the flagellum of a gram-positive bacteria, it only has 2 plates. The basal body has only 2 plates, and these plates are embedded in the inner cell membrane, which is a normal cell membrane of any cell. There's a cell wall here. The cell wall in gram-positive bacteria is quite thick, but overall, the flagellum has the same structure as we saw in the gram-negative. That there's a thread-like structure, which we call filament. There's a hook, and then there's a basal body. This is another picture of a comparison between the two bacteria. The flagellum of the 2 bacteria look like this. As you can see here, this is a gram-negative bacteria that has inner cell membrane, then there's an outer cell membrane. And in the sandwich between the 2 layer is periplasmic membrane and as well as cell wall. And as you can see, there are 4 rings in gram-negative bacteria, and there are only 2 rings in gram-positive bacteria. Gram-positive bacteria has a quite significantly thicker peptidoglycan layer. The flagellum consists of the filament as well as the hook and the basal body. And this basal body basically rotates. It can rotate clockwise as well as anti-clockwise. For normal motility, movement, it rotates anti-clockwise. Now, the motility or the movement of the bacteria could be demonstrated by using a slide that has a depression in it. It's called a cavity slide. It has a depression engraved on the slide. What we do is we take a broth culture. Broth culture is a liquid culture of a bacterium. We grow the bacterium for 16 hours or 18 hours. It is called a fresh culture. And then we take a drop of that fresh culture in a liquid medium. We put that drop onto a cover slip, and then that cover slip is everted onto this cavity. So if you look, that drop from the side, this is the side view. This is a depression here, and this is a cover slip. And that cover slip has underneath a drop of medium containing the bacterium. We can put this slide assembly under the microscope and can see the movement of the bacteria. So bacteria would be moving you would see them under the microscope moving from one side to the other. They're just random movements. For example, E. coli is a bacterium present in the fecal contents. It can revolve or make turns, 270 turns per second. So it's enormous movement. Bacteria can move with respect to 2 stimuli. It could be chemical, which we call chemotaxis, or it could be photo, which is light, phototaxis. if the bacteria senses that somewhere in the medium or in a swimming pool, for example, there is some chemical that it can use as a food. So it can sense it and then can move for the food. So that is called chemotaxis. And phototaxis is governed or dictated by the light. So light is also a signal for the movement of the bacterium. If the bacterium likes the light, it would go towards the light. If it hates light or doesn't like the light, it would go away from the light. The flagellum or flagella could also be used as antigen. The antigen that we name to the flagella antigen is called H antigen. And this H antigen, it can, if you inject this into a rabbit, for example, rabbit body would produce antibodies, and those antibodies could be used for identification of gram-negative bacteria. Because flagellum, which is a protein, it varies from species to species. So based on this antibodies that are specific to, say, for example, in this case, E. coli, we can identify E. coli from other gram-negative bacteria like Salmonella. Now, flagella, as I mentioned, it moves counterclockwise for forward thrust. So if it has to go forward, the flagellum would rotate counterclockwise. And there are two kinds of movements. When it rotates anti-clockwise, it runs. And when it stops, it rotates clockwise. So there are two movements. Run is an anti-clockwise movement, and the stop, or what we call tumble, is a clockwise movement. In this slide, it's a video clip where you could see the bacteria moving under the microscope. The slide, the cavity slide that I mentioned earlier, it is the same slide, and the picture has been taken from using the same cavity slide. So you can watch now. We can see the bacteria moving under the microscope. This is a video clip that shows it. As you can see, the bacteria moving very fast. This is a rotation. And here, look, you can see the bacteria moving. And the movement is pretty fast. Look, this one seems to be kind of stuck there, but still moving. And as you can see, the bacteria, these are two bacilli, rod-shaped bacteria held together, but still moving. This is a schematic diagram that represents rotation by the flagella. So this is the flagellum. And as you can see, the basal body basically here, the movement is happening here. And that rotates the flagellum.